what I'd like to then talk about would be these two topics, cloning, the cloning of humans, and the creation of life. So on this subject of the cloning of humans, which they say is very likely to happen, and when they say likely to happen, they mean likely before 2050, so in the next 40 years. When we pick up this subject, when you pick up the subject of human cloning, it has a long and colorful history, some of it science, some of it science fiction. But the topic of cloning, human cloning, really changes around the mid-70s, in particular in 1978 with the birth of a baby girl named Louise Joy Brown. So Louise Joy Brown was the first person to be born by in vitro fertilization, so the first test tube baby. And that was a very successful experience. Louise Brown had her own first child in 2006, so this girl has grown up to be a happy woman and mother. So that was a successful experiment, because when you're the first, you are at a certain level in experiment. And lots of people were very nervous even about this idea that now is somewhat common, this idea of in vitro fertilization or test tube babies. So for many years after that, it was still very much experimental medicine, but by the 80s, test tube babies, in vitro fertilization babies were being born around the globe, and governments were starting to wrestle with what kind of regulation would you put on this new technology. So that's 1978, but then in 1996, 97, another thing happened that's related to this idea of human cloning, which was Dolly the sheep was born. So Dolly the sheep she has passed on to sheep heaven. Dolly the sheep was a true clone. So when we're talking about clone here, we're talking about creating an exact copy as much as possible of an organism that has already been born. So Dolly was the proof in principle of a technology that's called somatic cell nuclear transfer. So what the idea is you take the nuclear material, the nucleus, and all the chromosomes from the animal or person you wish to clone. And then you get an egg from that same species, and you throw the nucleus out of the egg, and you replace it with the nuclear material from this other animal or person which you wish to have a clone of. Now this is pretty understandable as a technology if you want to do it with farm animals, because perhaps you have a champion milking cow or a champion bull or whatever animal you have that you really would, for financial reasons, want an exact copy of this animal. So this Dolly somatic cell nuclear transfer cloning technology could allow you to produce an exact copy of your prized farm animal, or perhaps if you were into racehorses, perhaps another northern dancer or whichever. So as a technology for producing farm animals, it seems like possibly a good idea, except that the technology has proved to be very inefficient and very difficult to duplicate in some animal species. So we can do it relatively easily in mice, and we can do it in some farm animals, and dogs and cats have all been cloned. But what turns out to be the problem is, first, it's very inefficient, and second, a good portion, say 25% of the animals that are cloned, don't do very well. They have significant anomalies. And so the idea of cloning a human person at a technological level, while this technology worked for Dolly in 1996, 97, people working on it for, say, another decade didn't make much progress with trying to figure out how to do it really efficiently with all mammals, and in particular with humans. So the big thing that would stop us from moving forward right now, even though they say it's quite likely to happen in this Scientific American article, is that we don't know how to do it yet. So it would just be an experimental process right now, and even in the animals where we say we can successfully clone them, we get about 25% of them that are significantly handicapped or don't live at all. So as a technology, we still have a long way to go. And then on top of the technology, well, it might be clear why you would want to reproduce your champion milking cow. It's not quite so clear as to why you would want to reproduce an exact copy of another human being. So normally, the old-fashioned way that babies are born, there's a way in which parents always know that the baby, the child, is going to be a surprise. It is the result biologically of the union of gametes from two different individuals, and we always hope that that occurs in the context of a loving family relationship and stuff. So you hope that the child will be received into a loving home, but the parents know that the child will be at all sorts of levels a surprise, and not just how long the child will keep you up at night, but that you don't know what this child, who's the product of a biological process at the physical level, is going to turn out to be. And so parents are ready for surprises. Maybe not all of the surprises, but they're ready for surprises. If you were trying to clone another human being, it seems that part of your intent would be to cut down on the surprises. That is, to produce a human being that is perhaps an exact copy of you, because you thought you should get to live forever, so it would be a way of producing a kind of immortality for yourself without having to become a vampire. And so you would have very definite expectations about what this child should be. He or she should be you, or perhaps even more dangerously, a better you. Somebody that you would hope would achieve some of the things that you didn't manage to achieve. Being a parent and knowing how to encourage and to nurture a child is a difficult process as it is, because you know, while you want to give the child opportunities to grow and opportunities for a rich life, you also know that this is a separate human being, and you want them to make these choices in freedom. So a child who is a clone, the freedom idea that the parents bring to that is likely to be much less than is healthy for either of the family members. The other thing you might be wanting to produce the child for would be to replace someone who you have lost, a lost spouse or another child who has died. And the idea of producing a replacement human being, while understandable that people would want to reignite, rekindle a lost relationship, we would be sympathetic to that, but to go about it in this way doesn't sound like a particularly healthy approach to things. And so the philosophers remind us that one of the sort of foundations of Western philosophy around our understanding of human persons has long been this idea that human beings are to be an end in themselves. So human beings are understood as unique, to become who they will become, and not as a means to the end of someone else's desire. So human beings are to have, I think we understand them as having a kind of inherent freedom that they are to discover who they are as opposed to being pre-programmed. And so that's why we, at this stage of human civilization, are generally find slavery repugnant and often also are very derisive of concepts like class structure and stuff, which say you are born into a particular place and that's who and what you will be. And on a night when we celebrate International Women's Day, we remember that we've come a long way in understanding a woman's role too. I think there was a time not too long ago when men's roles and women's roles were a lot more fixed. And I think in the last 40 or 50 years, we've come to say that while men and women may have particular unique traits, that we should in general encourage people to become that whatever they want to be. So that unique person, man, woman, race, creed, nationality, that we value this idea of allowing people to be free enough to discover what they have the skills to make them happy individuals. And so at a religious level, we would say the glory of God is a human being fully alive, and fully alive to be whatever you will be that will make you happy. The church enters this debate on human cloning very similarly to where the philosophers enter the debate that I mentioned, that all human beings are an end in themselves and not a means to some other end outside of themselves. We may choose to become something or to give our lives to something, but it is generally understood that that's a choice we should make. The church is also nervous about the in vitro fertilization technology and the test tube component of anything that would be, any process that would be involved in creating an embryo outside of the womb, and then of making the selection of which embryos we keep and which embryos we discard. And in fact, in the in vitro fertilization technology, that would have to be a significant component of any idea of cloning human beings. It is this idea of possible spare embryos that the church would want to speak very strongly against, to say, you know, the idea of creating a human being as a spare is problematic in itself, and so it's related to this idea of creating humans for an end other than themselves. And so some of the philosophical and religious concerns on human cloning are very similar, and for that reason, most social and religious groups have come out pretty clearly against the concept of human reproductive cloning. So the United Nations has a declaration, a non-binding declaration, but a declaration that it is against human reproductive cloning. Where this idea of cloning has gotten a little bit more complicated is as it relates to stem cell technology. And so now the cloning conversation gets divided into reproductive cloning, which I think most people, philosophers, the legal community, and politicians and political organizations are against, to this other thing that they call reproductive cloning. That they call therapeutic cloning. And that's related to the stem cell debate. And it's worthwhile knowing those terms, but tonight I don't think we have time to go too much into it. I just say that the stem cell debate is a debate which has moved also very quickly in the last dozen years, and it's now moved into a very interesting new phase where the research on embryonic stem cells, or stem cells that are like embryonic stem cells, has moved to a phase where it no longer requires the destruction of embryos. And that changes the whole conversation at the level of the ethics involved in human therapeutic cloning. So, but that's a whole other conversation. But I say that because it's worthwhile realizing that whenever we get talking about these kinds of issues that involve science, society, and communities of faith, it's important to realize that they're really complex issues to start with, but they're constantly changing environments. So this stem cell debate, it's related to the cloning debate, 5 years ago was in a very, very different and more contentious place than it is right now. So whatever you learn this year about these topics, try and learn a little bit more next year, and whatever you feel the challenge is to your religious understanding of who we are, living in community under the eye of a loving God, you will need to keep revisiting that, because this technology and the solutions it provides for questions of human health and the challenges it provides for questions of human, social, and religious ethics, these things are going to be constantly moving. So what this conversation about science, faith, and society, I think, constantly says is it's a conversation that we need to be engaged socially, just like we need to be engaged politically. That we can't, it's not enough for us to say, do something like vote every 4 years and say that's what democracy is. Well, it's not enough for us to think about these issues once and let it go. And the payoff is that I think when we're thinking and trying to learn about what the science is doing and proposing, we're learning about new technologies that will require important social decisions about how we will use the technologies. When we're thinking about the religious implications, it causes us to try and understand who we are standing before our God, and to try and understand how it is that God is active in our lives. And those are all very good things for us to spend some time on. Okay, I'd like to move to the second topic, which is listed on this list as the creation of life. The other name by which that topic goes is synthetic biology. And I'm more familiar with hearing this used called synthetic biology. And there was actually a big breakthrough in this area this past year. So depending on how you define the creation of life, this very likely to happen thing happened in June. And in fact, not only was it in the science journals, but it was on the cover of Time magazine, and there was a really nice cartoon version announcing this creation of life story on the front of The Economist magazine the week that this was published. So the idea of the creation of life or synthetic biology, what that's all about is really the front edge, the leading edge of what we have for a long time called genetic engineering. So what genetic engineering allows us to do is to isolate individual genes, to study them, to see more about their structure, to see about how the DNA is coding for the proteins, and then how the different genes are regulated and cause different cells to be what they are. So molecular biology has given us these tools of gene cloning, and we've been doing that since the mid-70s. And so what this creation of life idea is that can we actually synthesize a whole genome rather than just parts of a genome, single genes or even chromosomes? Can we synthesize a whole genome that we have designed and cause that to produce life? Well, we're not there yet, but what happened last May or June was a group in the United States headed by a character named Craig Ventnor, who's a very big genome project guy. He's the guy that was in charge of the privatized component of the Human Genome Project. And Craig Ventnor is always pushing. So what they did was they over the years have identified what seems to be about the smallest bacterial genome that we have that is able to live on its own, you know, in the proper soup. So what they did was design a copy of this bacterial genome in their computer, and they put special little watermarks, little special signatures in it that shouldn't disrupt the gene function of the genes in this genome, but would allow them to tell the one that they've designed and created from the wild type one. So what they did then, so they designed your genome on the computer. So the idea here is you go from an idea to a genome that you've designed on the computer, and then you try and then you have to produce that genome, and then somehow you have to make that genome make a bacteria alive on its own, or make a cell or an organism alive on its own. This is in very early stages, so they designed their genome, had it produced, so using the absolute best techniques in molecular biology today, they produced a copy of this single bacterial genome, which was basically 1 million base pairs long, 1.1 megabases, so around 1 million base pairs long. When I first started cloning genes around mid-80s, so in 1987, I was involved in a team with the first time I was cloning genes. Our gene was 18,000 nucleotides long. This is 1.1 million long. So 18,000, 20,000, 50,000, even 100,000 we've been able to do for a while. But 1 million, to be able to reproduce in a test tube by chemistry, a genome of that size is quite a technical feat all by itself. And they did some neat molecular biology tricks to do it. So the real gold standard now is to take this genome that you have produced yourself by chemistry and make it come to life, make it become the blueprint for a living organism. Well, they haven't figured out how to move from chemistry to biology yet. But what they did in this experiment last year was they took a bacteria with a genome very similar to the one they've designed. This was the blueprint for the one that they modified. And they find a way, it's a fairly standard molecular biology technique of introducing this new genome into the bacteria that's already there. And then by various drug selection tricks, you can cause the genome that you're interested in to survive and the parent genome to disappear. And that's what they did. So this was the first time that you had a genome arise that came from somebody's mind and someone's computer rather than from evolution. Now, it cheats in the course. If you were trying to do this with God, he'd say, get your own dirt, you know. Here, we've copied the genome that already existed, but we've played some tricks with it so that we know that the one that is reproduced in the bacteria several generations later is the one that we made rather than the one that Mother Nature had designed. So that's quite a trick in itself. It's not quite producing life from nothing or even producing life from chemistry, but it's a pretty darn good trick. We haven't done it before, and they're working with massive pieces of DNA, and that's pretty darn good all by itself. So why would you do this kind of thing? Why would you want to produce your own design of a particular bacteria? Well, a lot of biotechnology is geared towards producing important biological processes for medical uses. So to produce insulin more effectively and efficiently, to produce growth hormone, to produce products like interferon that you might use for treatment of viral diseases or tumors. If you can engineer bacteria that will do this job for you, then you can put them in fermenters, and the same technology we use to make beer, we can use to produce biological products that are active and relatively easy to purify. So those are the kinds of good things that you can do with it. I guess one of the other things that people bring up is this idea that maybe we could produce bacteria that would eat oil, and so when you had an oil spill, you could spread this bacteria on the oil spill and it would chew it up. But that use of that technology points out one of the problems that people worry about. Well, it's okay if you produce a bacteria to do something that you want if you can control it, or if the bacteria doesn't do anything you don't want it to do. And because we're copying Mother Nature and we're playing an evolutionary game, when we make these changes, we don't exactly know everything that that new organism might be able to do. We may be selecting it, designing it, and selecting it so that it can do certain things, but there may be certain things that would surprise us that it would do that we would like to contain. And so the idea of using the bacteria in a fermenter where you contain it and where you destroy it when it's done what you created it for, that doesn't make people so worried. But the idea of putting it out in the environment like the ocean to chew up an oil spill, while we might be very happy to have the oil spill gone, we're not sure what it would go and do to the environment after that. So that's a problem, and people would are also worried in our day and age about questions of bioterrorism. Now there's some good old-fashioned bacteria around that are perfectly good bioterrorist tools, but you know, something like anthrax, which people have played bioterrorism tricks with. But the idea that you could create, you could develop something that would have a very nasty bioterrorism capacity makes people very worried. So very, very shortly after this paper was published last May or June, President Obama in the United States set up a committee to study the pros and cons of this synthetic biology technology, and to report to him in 6 months as to what sort of regulatory activities that we should include in the way we handle this, this new technology. Because people are aware that this is a powerful new technology and that it is likely to need to be regulated. But that points out a problem. The technology is moving very fast. So President Obama strikes a committee to look at this technology after it's already published and out there. If we back up to talk about the question of human cloning and in vitro fertilization and stuff, I said that Louise Brown is born in 1978, and by the 80s, there are a lot of children being born by in vitro fertilization. The Canadian government started its Royal Commission on New Reproductive Technologies in 1990. So a dozen years after the birth of Louise Brown. The bill for the new reproductive technology was passed in 2004. And not just because government moves slowly, but because these technologies keep changing so much that they're hard to regulate. But they're so powerful that people widely believe that they do need to be regulated. With respect to technologies like gene cloning and synthetic biology, the Church's position seems to be fairly general and basic, which is that gene cloning technology isn't of itself a problem, it's the use to which we put it. And so if the technology is put to use to help human beings, to help with environmental problems, then in general the church, I think, would be quite positive about it, but would have the same qualifiers that other regulatory agencies would have, that you really do have to be careful. These are very powerful technologies, and if you're thinking about something like a bacterium, once you release it into the environment, if it's able to reproduce, it can be very difficult to recall. So that if you're thinking of releasing these organisms into the environment, it would be good to have some sort of genetic fail-safe program built into them. So I think, I would say in general, the church and governments are open to this technology, but want us to recognize how powerful it is, and therefore to use the kind of care that we should use with it.